Good evening from San Antonio. I'm Jody Shoger, and I'm here with the Alamo Breast Cancer Foundation. It's a pleasure to be a scholarship advocate this year, and I'm pleased to take part in this conversation.
My name is Chang. I'm really pleased to be here and to answer some of the questions that we may have. I'm a professor at Royal Cornell at the Methodist Hospital in Texas, Houston.
Well, it's great for you to be here, Dr. Chang. This has been an amazing conference, and I think one of the things we want to start with is, from everything you've heard, I think there's, 1,200 abstracts, something like that?
Yes.
But what have you found the most compelling that you want to take back to your patients?
I think that, we're in a genomic era, and what we've discovered is that not all breast cancers are alike. They're very heterogeneous. Like today, the whole morning was spent on the different types of how could we select the best therapies, was it on PI3 kinase, for HER2-positive breast cancers. And I guess the most heterogeneous cancer of all, I think, would be triple-negative breast cancer, for which there are many subtypes. And again, a very elegant paper that was done in collaboration with Foundation Medicine, the Vanderbilt Group, looked at residual tumors after neoadjuvant chemotherapy and came up with the mutations that were present in triple-negative breast cancer. So I think where we're coming from now is that cancer, especially breast cancer, we understand it's not just one disease just because it arises in the breast, and that different women respond differently, and how do we select the right treatment for individuals. And that, to me, is very exciting and very difficult, because now you're looking at very small subsets of women that we can actually give therapies to. So how do we next design the next generation of studies? Do we test all women with all new drugs? Or how do we learn to select, and how can we be clever about these mechanisms?
Did you hear more about that selection process for the different subsets?
Well, other than being smarter and doing next-gen sequencing, etc. I think one of the more compelling talks that I heard, again, was from Dr. Lewis Kentley, and he was a plenary speaker, and he has designed something called the cold clinical trial. And I thought that was extremely exciting. And so what that is that instead of testing new combinations of drugs, can we test these combinations of drugs, because it's going to take combinations. Can we test it in an animal, in a mouse hospital, as a co-clinical trial, a clinical trial conducted in mice that have the genetic background that imitates our patients, so that we know that instead of testing combinations that may not work, not all combinations do, but can we test it out in the genetically modified mouse or a human xenograft mouse, meaning a biopsy taken from patients into mice, and to figure out who is most likely to respond. And he gave great examples, which I thought was very, very compelling and very exciting to move into the next phase of triple-negative breast cancer research.
Oh, that is exciting. And that is obviously a way that targets might be identified and drugs developed in a quicker fashion, do you think?
And tested in the right population to the patients, so that instead of doing thousands and thousands of patients, we can do it in smaller numbers of patients with a positive result.
So is this one of the studies that you'll take back to your patients that you work with? I think to talk with them about?
Definitely. And also to how I'm going to how we move forward as a collective whole in designing the right clinical trial based on scientific rationale, hopefully based on preclinical animal hospital co-clinical trials, and to find out the best combination to give our selected patients to individualized therapies for maximum success.
That's excellent. That's excellent. Do you want to talk to us a little bit more about your work?
We do a lot of work in therapy resistance and metastases. Do a lot about and what are the drivers behind therapy resistance and metastases, especially in triple-negative breast cancer. And finding drugs that are effective in triple-negative breast cancer that are refractory to conventional therapies. And so we have obviously a number of PI3 kinase trials that are ongoing, and a number of reposition drugs that are ongoing in terms of clinical trials. We do have an animal clinical trial. We have an animal clinical facility, a co-clinical trial or animal hospital.
An animal.
And so I want to be able to scale that up and get more drugs into that to basically find out how we give treatments to women that may benefit them.
Excellent. And there have been a lot of headlines this week, and I'm sure there are a lot of phones ringing in doctors' offices. And can you tell us a little bit more about what you thought, for example, about the chemo brain study? Have you had time to look at that, or the Atlas results and what that means?
So I do think that the Atlas results is a 5 versus 10 years of tamoxifen. It's obviously very, very important. And it basically is a positive result, to basically summarize it, that the longer endocrine therapy is of benefit. And I think that most people have already, especially for higher risk patients, begun to practice a longer endocrine therapy.
I see.
So whether you stop at 5 years, or give after 5 years of tamoxifen in a postmenopausal woman an aromatase inhibitor, I think that is generally what most practitioners have been doing. And so I think the Atlas was a heroic effort. And I remember when it started, I was a fellow in the UK. I remember it starting at that time. And I think it's a great effort. And it does show that prolonged endocrine therapy will benefit some women at high risk.
Excellent. Great.
It was a great session.
It was interesting, and a lot of evidence that works for a certain subset.
Yes, correct.
And but not everyone.
Not everyone. That's correct.
Not everyone. What else have you seen that makes you glad to be a doctor?
Oh, today's results. I think it brought a tear to my eye.
Oh.
But the results of all the 10-year results of the combined NSABP and CCTG Herceptin study, the trastuzumab study, that showed the very first targeted therapy that made such an enormous impact on the aggressive, lethal HER2-positive breast cancer, and how we have come 10 years down the line. And to the PhD, Dr. Brown, who basically said to these researchers, work together, publish the results together, and the late Dr. Brown. And I think that only if we collaborate, design the right study, do it together, are we going to basically make a success, as we have done in saving many women with HER2-positive breast cancer with adjuvant trastuzumab. And I think that is scientific collaboration, team science.
Oh, well said. Thank you very much. That was wonderful.